I guess we start. Won't get much more. Yep, let's see. Maybe we're extra productive if we're only a few people. So last week we tried to implement this modularity score. My original plan was to go on with that, but I spent yesterday evening implementing this, and well, I managed, but it took me much longer than I thought, and somehow I came to a conclusion that this is an annoying exercise, and maybe there's more productive stuff. So just to wrap that one up and then do something else, I show you what I've done. So this is what we've done last time, as you might remember. We had this matrix of neighbors where for each cell we had its 10 or 15 nearest neighbors, and when we wrote in this thing to make an edge list, which was a list of lists, which just tells us for each cell all its neighbors. And when we had this group variable, where each, I'm going to make this a bit bigger, where each cell is assigned to one group or cluster or community or whatever you want to call it. And we wanted to know which fraction of all the edges between cells, so of all the neighborhood relation, is actually inside one group of all the edges, and we want to know what would we expect if we randomly reconnect the edges. When I looked at this yesterday, I realized that this doesn't make sense, because here we just went through the edge list, and for each vertex, we just assigned its neighbors, so its 10 nearest neighbors, so each vertex has degree 10. But the whole point was that we said that in these neighborhood graphs, we can get higher degrees, because sometimes vertex A considers vertex B as a neighbor, but not vice versa. And so what was missing here was that I added the neighboring vertex to the edge list of a vertex we are looking at, but not the other way around. So I fixed that and rewrote this function, also changed the name from edge list to list of neighbor lists, LNL, because I thought that might be a better name. And now I just make this list when I go through all the cells, and then I go through all the neighbors of a given cell. So V1 is a cell and V2 is a neighbor of this cell, and then I add V2 to the list of neighbors of V1 and vice versa. And I always check that I don't have it yet in order to get both. Now, you might remember last time we already had this modularity score implementation from the iGraph package that we used in R. And we compared and checked that our code gives the same modularity score as the iGraph code. So we concluded that the code is correct. And now that I fixed the bug, I don't get the same number anymore. That confused me a lot. And tell you what the solution was that I realized after a while. In iGraph, a graph is supposed to, in math, you make a distinction between a simple graph and a multigraph. A multigraph is a graph where you can have more than one edge connecting two vertices. And in iGraph, it's assumed that a graph is always a simple graph. And if you try to add the same edge twice, it will not add it twice. But in order to make the whole thing fast, they don't check for that. You have to explicitly call a function called simplify, which goes through the whole edge list and kicks out all the double edges. Because we haven't done that, our code in R also had all these edges, didn't have proper pruning of the edges. And so we had the same mistake in both of them, in both modularity scores were wrong. So once I ran simplify on the iGraph thing, I get the right modularity score. Now, the rest of the code was correct. So here to calculate the actual fraction, we just go through all the vertices and all their neighbors. And for every edge that we see along this list, we check whether the two connected vertices are in the same group or not. And then count how often they are in the same group, divided by how many there are in total. And for the opposite thing, to see what we expect, we calculate how many vertices to leave from each, how many edge stubs are there in each group, by just going through all the vertices in the group and adding up the degrees of these vertices. And that's what this function here does. It just goes through the vertices and gives you back a table which says in group 1 there is 3000 edge stubs, in group 2 there's 200 edge stubs, and by an edge stub I mean an edge starting there, and even without looking at where it ends, whether it ends in the same thing. So an edge stub is half an edge. And then you can calculate how many edge stubs you expect, by just asking what's the probability that a vertex, that an edge leaving from a given group ends up in the same group. And for this you have to take the fraction of edges leaving from this group and square it, because this fraction is the probability of picking a starting point in the group, and it's also the probability of picking an ending point in the group. So if you multiply it with itself, you get the probability of picking an edge which is completely within. So that's the formula we already had last time, where we said we have to take this table of them and calculate them. And then the thing we wanted to do later, and which turned out to be a bit annoying, is if you then want to know what happens if I take a vertex and move it from group A to group B, how does the score change? And to this you have to ask how does this actual fraction change, and how does the expected fraction change? Because you remember our modularity score was the actual fraction minus the expected fraction. Here, actual fraction minus the expected fraction. And if you now move something, if I move vertex V from the group where it was to a new group, I can ask how do things change? And of course, I first ask how does this actual thing change? And for this, I have to ask, you remember that was just the number of internal edges. So if I move a vertex from one group to another, and I have to say all the connection from this vertex to the old group now vanish from the list of actual internal edges, because they are no longer internal, because we moved the vertex out, and all ones going to the new one go in. So that's what I do here. I go through all the neighbors of the vertices, and whenever I see the neighbor was in the old group, I say this is now an edge which I lose, which is no longer internal. And if the neighbor is in a new group, it's an edge I gain. So this is now internal. And this is why I go here through all the neighbors of the edge of the vertex to be moved and calculate how much it changes. And when I get here how the fraction changes is, of course, this delta divided the total number of edges. And the other thing, this thing with the other term, the one with the squares here, I have to check how does this square change. And of course, the old vertex gets reduced by 1, and the new, or gets reduced by the degree of a vertex, and the new group gets increased. So that's what I do here. I say, I take the old group, I take the number of edge stubs in the old group, minus the number of edge stubs which now vanish, which is the number of neighbors of this vertex. So this is old groups, and I square this and subtract the value that we have before. And here I do the same for the new group, and then I see here how the second term changes, and then I calculate how the actual thing changes. This then allows me to optimize and just go through all the vertices in turn, and every vertex here, I take a vertex. Here I did it that I always take a random vertex, and then I go through all the groups and always ask, if I move this vertex into this new group, will the score increase or not? And find the one where it increases most. And if I found something which increases it most, I move a vertex to the new one. And then I ran this, and what then happens is, here you see, now this was just debug info, and now comes the interesting thing. We start with a modularity score of 0.01 if at the beginning every vertex in its own group, and then it increases and increases while the number of groups goes down, because more and more vertices are put together in a group. Now it's surprisingly how many little bugs you can make when implementing something like that, especially trying to get this formula here right. So I thought, let's not go through that, but let's go through something else which might be more interesting anyway. And that has to do with the pseudotime that I mentioned on Monday at the end. We want to see how we cells manually change. So this is again with cells that we always had, with a data set from this adult neurogenesis thing, where these are the stem cells. And when the stem cells become activated and start to cycle, and then they become neurons. And the first thing we will have to figure out is here, these neurons, they should be connected here, but they aren't. And the second thing is when we want to put a pseudotime through it, which starts here and moves along and gives this thing more and more increasing values, where this gets values around 0, here around a half, and here around 1. And that's what I now want to do sort of on the computer. And next Wednesday or the Monday after, whatever, we'll talk about how to actually talk about the math behind that. So, and for this, what I wanted to do, in case you want to try along, is I took this data set that I had last time. It had 13,000 cells, which makes everything a bit slow, so I subset it down to only 2,000 cells. And with that, we can probably work better. So I have put this here on Moodle. You see here at the bottom. So maybe you can download this data set and load it in R. And I do this as well for a moment. So here is my code that I've done, and maybe I start at 0 and then upload the code so that you can quickly do the same. So I first go through the code and then you can upload it and see it. So I just load the tidyverse and I load the Sora package. Then I read my RDA file, which contains the count matrix, and I run this through our usual pipeline of creating the Sora object, normalizing the data, scaling it, running a PCA, finding the neighbors, running this Louvain clustering, and then making our UMAP plot. And if I do all this, I get afterwards this plot here. And so that and I think I quickly put this here, also update and hinzufügen, Datei, nicht Datei, Textseite. Maybe I give you for a moment to load this thing and then we can talk about it. You all have an R installed on your computer and a Sora. And tell me when you've got your stuff loaded and have a UMAP displayed. Yeah. What doesn't work? Yeah, the UMAP always looks slightly different. That's because in the UMAP you have a so the UMAP works the way that it starts by placing the cells at random on the 2D plotting surface, and then it does some optimization where it tries to push together cells which are neighbors. And this of course depends a bit on the initial state. And you can set a random seed to seed the random number generator so that it always starts with the same thing. And it sets the seed to 42. But it seems that this isn't platform independent. I think there might be a slightly different random number generator in the Linux and the Windows implementation of R, because I've noticed that it only looks the same if you use the same platform. In any case, to see whether yours is sufficiently similar, I have made here, you see, what we can now do, we can look here at plotting for aquaporine 4, which is with astrocyte and stem cell marker. So we see there should be one elongated thing which has the AQP4 at the beginning. And then comes the cell cycle, and I use the cell cycle marker MKI67. And we see these are the cycling cells. And the last one, double cortex, are the neuroblasts, so the cells which come out. So the way how the pseudotime actually works is we start here, go through the cell cycle, and then end up here. So as long as you see these three components, it should be fine. And at least on my computer, there is this gap, and it seems the UMAP has torn my lineage into two halves. Anybody? Do you all have that? The server package? Yeah, the tidyverse, okay. It's annoying. Good, while it's installing, when I tell you a bit more. So one thing, I might have shown you the Sleepwalk package before, where I put the mouse somewhere and it shows me the distance in PCA space to all, from the cell under my mouse cursor to all the other cells. And there you can now see all these cells which are now in dark color are close by. And I can move here, and now I can see this trajectory here. And then how it goes here and over there. And now I can see how this is connected. These things, these neuroblasts, they seem to be connected with that point here. If I go here, then you see this is connected. So this seems to be the mistake that UMAP did. It didn't realize that this cell here should be placed next to that cell, because these cells, they are clearly connected to these here. And that probably happens because there are so many other cells, and here there's such a weak connection with only a few cells sitting out here. That's because when the cell exits the cell cycle, they change quite fast. So what I imagine what's happening here is that the cells in a real space sit somewhere like this, and then and when they get so when they are sleeping, they are all more or less the same. When they get activated, they sort of change from the state space and become different to these cells, and then they become more and more different, and then they add the cell cycle, and then they jump quite quickly to become neuroblasts. And because this transition from the cell cycle to the neuroblasts is so fast that we only catch very few cells in this intermediate part, which we don't have enough cells for them to get connected. But if we probe the thing with this sleepwalk tool, then we can see that there should be a connection here, because we can see that this cell is very similar to that cell. So this is the cell which bridges between this and this. Now, the thing that I now wanted to do is, yes, you also have to install the sleepwalk package. Just say install package the sleepwalk. In R, when you use something like this here, with this double colon, it means take this command out of this package, even though the package isn't loaded. But of course, it has to be installed. So, how's your computer coming along? Okay, so the thing that I now would do next is I want to make this pseudotime coloring that I showed you at the beginning, which gives each cell a number which slowly increases from 0 to maybe 1/2 to 1 along this trajectory. And in order to do this, what I do is I take here my numbered clusters and say my lineage is 7, 1, 3, 5, 4, 0, like this here. These are all the clusters in the lineage, and the other clusters here, 2, 9, 6, 8, they aren't part of the lineage. So I take these, and what I now do is I take my PCA coordinates. So the PCA transformation, here you see each cell now is written here by, described by 20 principal components, or 16 it seems, okay. And so this is the result of this run PCA up here, gives me these ones. And I just ask for each cell, here is what the cluster is for each cell, and here this way I ask this to tell me which cells are in these clusters and in the others. So I subset, and now I have here in X a matrix which gives me the 20 PCA coordinates for the 1600 cells. For the 1600 cells that are in this that are in this lineage. And now there's this print curve package, and what this thing does is if you have points which simply sit somehow like this, it draws a line through them, and then gives each cell a position on the line by projecting it on the line, and then calculating the, and then also giving it a number which tells us how far along the line is. And I've done this here. I used this print curve library and now tell it to calculate a principal curve. And then I display this principal curve here. And as we can see, it does something slightly strange. It starts here, goes up to here. Now it jumps over there, and then it continues here, which we will need to rectify in a moment. And the reason why it jumps so much is, if we look at Sleepwalk, you can see that somehow this here is rather close connected to that, even though the UMAP doesn't show it properly, while these cell cycles here, they are really far out. If I go onto the cell cycle, I don't see any of the other cells. So what I actually have here is, I have here a blob of cells which are the stem cells, and then they get activated, and then they enter the cell cycle and go very far down here, come back from the cell cycle, and then they continue here to the neuroblasts. And now if I tell it to make a curve through it, it makes the curve like this instead of like this. So we might want to remove all this part here. But before we get into this, I want to do something else. I want to show what actually this principal curve package does. And it's hard to see in this PCA space because the PCA space is so well, it's 20-dimensional. We can't visualize it. So let's use the PCA to make a curve in 2D. And for this, we start with a curve, and then we put noise to the curve, and then we see whether we can get back to the curve. So let's try that. What I now want to do is, I want to just draw on my screen some curved thing. Let's say, let's start with simply a sine curve. And then let's put points around it which sort of scatter around the sine curve. And the question is, how can we construct example data with x and y coordinates so that they follow such a sine curve? Now you can try yourself, or who has an idea how we best do that? Any ideas? So let's first draw a sine curve onto the screen. And the way how I could do that is, usually one possibility would be this. I just show you, and then we can see sequence 0, 2 times pi, comma, length out is 1000. Now you see this here is a sequence of values which just go from 0 to 2 pi. And if I now plot something like plot t, sinus of sine of t, then I get my sine. The way how I now want to do that is, of course, here we already have a curve and we want to obscure the curve. So the first thing that I do is I get a couple of, let me see. That's actually, you know what? Let's make a spiral. From 2 to 4 pi, plot t cos t times cos t times sine p. Now we have here a spiral. You see how this works? We start here and go here and here. Let's make it a half spiral. I start somewhere at 2 pi and go to 3.5 pi. So this is now a nice curve which does a bit a non-trivial thing. It's also not too complicated and gives me points on this curve. But I want random points. So what I do now is, instead of making t like this along this sequence, I say t is a uniformly distributed number of 1,000 points, uniformly taking from this area between 2 pi and 3.5 pi. So these are now these numbers. And then I can say x is and y is that. And if I now say plot x comma y, then you see the plot still looks like this, but now it's slightly more, now it's slightly uniform. There's a few points missing and a few more here. So this is now how I made my example curve. I just have here some plot, here some plot, and now I tell it plot that. But we wanted the whole thing to also be a bit noisy, that it scatters around a bit. Has anybody got an idea how to add scattering to that the points don't lie exactly on the line, but slightly next to it? Yep. Okay, let's add a random number. What random number would you use? No, where's my plus here? Yeah. Okay, so in R we have this R norm function, which allows us to add random stuff. And I can tell it add 1,000 random numbers with a mean of 0 and a standard deviation of how much should we take, maybe 1 or something. And for the sign, I do the same. And now it looks like this. I add here this aspect ratio 1 so that this x and y axis is scaled the same way. And now let's see whether our principal curve package manages to follow this, to recover this curve here. So what I now do is I use this print curve function, and what the principal curve function wants in the easiest case is just a matrix with x and y coordinates. So I tell it x comma y like this here. You see this is now just a long matrix with the coordinates next to each other. And if I call this, then it gives me my principal curve. And we can now look a bit at what we have in this principal curve. PC. So there is here, the first thing is this lambda. That's the number that each cell gets in this principal curve. So, and we can see whether this has worked. I do the following. I say tibble x comma y comma, and here lambda is PC$s. So this is this s thing that the function has given us. And if I now plot this here, first, if I just plot it like this, I get the plot we just had. And now I can also, I could, for example, say color equals, let's add here also something like mutate index gleich row number. So what I now have, you see here, is each row now gets a number, and I can color by this number. And if I do that, then what you can see is this is the cells as we have them in our list, and they're in a completely random order. And now our principal curve package has now assigned each cell a number S, which now brings the thing in the proper order. Right? What did I call it? Lambda. Why did I call it lambda actually? I wanted to call it S if it's also called S here. So here this is our stuff and S, X, Y, because it is, sorry. Yeah, I wanted to use this lambda here, which it gives us. And you see the lambda now correctly gave numbers from black to light, from 0 to 40, going along this thing. So it has correctly managed to make this line here around. What it has also done is that it gave to each, that it has also produced is this $PC$ here, which gives us for each cell the closest point on the line. I can show you this plot. And you see, this is the curve that it has reconstructed. This was our original curve. Well, this here in a way without the noise would have been our original curve. And this is the reconstruction. Plot. And we make here a lines $S. Alt, this doesn't work. We see $ort. Up, comma, I have it in a moment. So now what you can see here is the black line. The plots here is our data points, and the red line is the curve that it has constructed. It's not exact. Our original curve was here vertical and here horizontal. It hasn't managed this, but it got quite close to that. And this is now, and this now shows you how this works. The principal curve package, it works by making, by finding this red line, which follows the points as close as possible. And as close as possible means every point, every cell is now replaced by a point on the red line, and the distance from the cell to this red line is minimal. So it calculates, for each cell we now have two coordinates, the coordinate where the point was before and the coordinate at the closest point on the line. And $S is this closest point on the line, and that's how we now get it. Should maybe make this like this when it's more obvious. And you can now see how each point is replaced by its line point, and then you get this line. And it just tries different curves and tries to minimize the squared distances of each point to its closest point on the line. And we need to know what kind of distances it tries and what kind of curves it tries, and that's what I wanted to explain next lecture in a bit more detail. But for now, we just see this thing and we can play around a bit with it. And see what happens. We can also maybe we can now try things like see how does this thing actually react if we make a gap like the thing that we had, like the issue that we have here with the big extra step for the cell cycle. By so this is two-dimensional. Let's see how we can, if we do this nicely in 3D. Now let's now go back to our original server object, and now that you see how the principal curve package has worked in principle, maybe now that we're here, I show you a bit how this thing works individually, because what it does, it tries to, I mentioned this a bit before, and And let's try it again. What it does, it always flips back and forth between bringing the cells in the right order and making the curve. So at the moment, each cell now gets a position which tells it where it is. And I can make plots like plot PC$S, that's the number that the cell has along this line, so this pseudo-time thing, no, lambda, sorry. And the x coordinate. And now you see this is our sign where we have here the x coordinate and here the cell. And once we have this, it's now easy to draw a smooth line through that by doing so-called smooth regression. Here we use spline regression, which works the way that we try to reconstruct this thing as a linear combination of a spline basis. A spline basis looks like this. And we have it. Which picture? Let's take this picture here. This here is a typical spline basis where you have now a couple of 1, 2, 3, 4, 5, 6, 7, 8 functions. And for each of these 8 functions, we can make a linear combination. Let's quickly try to make this spline basis. In R, this is done with the function bs. So if I use this bs, I can give it, I make me a grid sequence 0, 1, length out is 1000. And now I can make BST, library splines. And we quickly try to, and as you can see, this BS function has now produced me a matrix with 3 columns. And to understand what's happening here, I plot my T versus the first column of this matrix. And as you can see, that's this function here. This is the second column, map, and this is the third one. There's also a fourth one, which I get only if I say here intercept is false. So string x. So I tell this BS here something like, let's call this B. And then I can now here say plot T, B at 1 gives me this. Sorry, intercept BS. If true is included, true, I want an intercept. So this is my first basis line, this is my second basis line, this is my third, and this is my fourth. So it gives me 4 different curves. I can also, if I tell this here, degrees of freedoms is 6, then hat B gives me 6 columns, and again, they give me these colors. First column, second column, third column, fourth column, fifth column, sixth column. And each column is a plot, and the first column is a curve which peaks to the left, the next one a bit more to the right, the third one in the middle, the fourth one a bit further to the right, and the last one to the very right. And so I can now build up different functions from that. For example, I could take say, let's take the first column times 0 plus the second plus the third plus nothing of the fourth, fifth, and sixth, like this. You see, this makes a linear combination. So this is matrix vector multiplication, multiplying the first matrix of a vector with 0, the second column of a matrix with 1, the third with 1, and so on, and adding this up. And if I do that, what I get is now a function which looks like this. Or if I put 1 on all three of them, it gets wider. Now it gets even wider. Maybe I can make here a 0.5 when I get maybe a dip. So you can imagine how I can make different curves by putting different coefficients here. And you can play a bit with this if you want to see how you can make different curve shapes by playing with these coefficients. And that is what spline regression does. So let's write this here, a grid of values from 0 to 1. No, this hasn't worked. Not 0. Ah, my T is wrong. So I've now written code here while you were looking at your own stuff, just putting all these 6 basis planes in one picture. And you see first, second, third, fourth, fifth, sixth. And then you can imagine here, if I take 0 times this, plus 1 times this, plus half times that, and 1 times this, and 1.5 times, then of course I get a curve like this. And the funny thing in, everybody with me so far? Now, the nice thing is, what we can now do is, if you remember, we had here this plot, and we want to make a, and we want to find a spline which follows this. And what we can now do is, we can make a regression by saying PC $x tilde BS PC $lambda, comma, intercept is true, comma, df is 6, plus 0, So now what happened here is quite a nice piece of magic. I have here my random data where all these x and y values follow a curve, but they wiggle around the curve, and I want to have a smooth curve that follows them. And what I now do is I calculate for each of the lambda values, for each of the x values here on my plot, I calculate the corresponding value of these 6 spline curves. So you see here, for each value of that I have on the x-axis, I can look up what is the value of all 6 spline curves. And if I do that, here I get here again this matrix, but now the different rows don't correspond to the linear grid going from 0 to 1, but each row contains to the pseudotime value of one cell. So here in this plot, to one of the x values of these points. And here is the corresponding y values of the 6 different basis spline curves. And we now do a linear regression by asking R, find me 6 coefficients like these here, such that the value that we produce, that the y value we get by assembling them, gets as close as possible to what I have here on this axis. And this is what, and when I get my 6 coefficients, these ones, and for the first, second, third, fourth, fifth, and sixth, and if I plot it, I get that. And this is called spline smoothing. I have a scatter plot of values. I have a scatter plot with values, and I calculate my spline basis values for each x position, for each x coordinate, and then try to regress this by saying, now find the 6 coefficients such that the fitted y coordinates, so the red curve, gets as close as possible to the black curve in a least square criterion. And the LM function that I've done here is the completely normal ordinary least square function. Because what you see here, what we are here doing is we have this thing and say each y value should be produced as a linear combination of these 6 values, and the linear combination should be chosen such that the linear combination of these 6 values gets as close as possible to the y values, with as close as possible the usual least square criterion. Hence, in order to solve that and find the coefficient, we also use the usual least square solver, which you might remember is essentially a matrix inversion. Everybody remembers how least square solving works? So if you have something like so what we have here is for each y value, we now say we want the x value, we want to say form it by these 6 x values, which I call beta 1 x1 plus beta 2 x2 plus beta 6 x6. And I can, of course, also say this is a vector x multiplied by this vector beta. And I can say the whole matrix Y, the whole data matrix, the vector of all the data values, is then given as this matrix X times beta. So this X here is the matrix of all these BS values, the spline coordinate, the spline values for the different values of our t. And I multiply this from the left with this beta, and then I get my y. And what I now want is that this value that I get that way minus y hat that this gets minimal. So I want to find the betas, that I want to find those 6 values for beta, that this value minus the real y value is sum over all I xi beta minus yi squared is minimal. And in order to do this, what we need to find is there is a way to invert the matrix, which is very similar to how you solve a system of linear equations with Gaussian elimination. You might remember in high school where you do this, add one equation with the other and then bring it in this triangular form so that you can interpret backwards, you know what I mean? And yeah, I haven't, I should have made a no, why does it talk now? You might remember this from high school. You have some equation system where you say 1 times x1 plus 3 times x2 plus 1 times x3 plus 9 times x2 is 0. And 1 times this, and when you do this, where you say, okay, I have to multiply this thing by 2, by minus 2, and then add these things, and in that way you get the whole thing in some form that we have all zeros in front. And it turns out that in order to find the optimal solution for the betas, you do exactly the same. I don't want to get into these details now because I'm not sure I can explain it now in general, but this is your standard effect whenever you do a linear regression where you do something like like some dependent variable depends on several independent variables in a linear way, and you want to find out the coefficients when you use this method. And in R, it's this function LM for linear models that does it. So what I wanted to show you is, as you can see, wanted to explain what the principal curve function does. You remember we started off with this curve here, and then what the principal curve function now needs to do is 2 things. First, it needs to bring the points in the right order. And once it has the points in the right order, that it knows this is position 1, this is position 2, 3, 4, 5, 6, 7, then it can plot now. It can now plot sort of this order here, which I call lambda. Lambda starts here and increases up to here. And then we can plot the x coordinate against lambda and the y coordinate against lambda. And here you see the plot of the x coordinate against lambda, and here you see the plot of the y coordinate against lambda. And in both cases, I can make a fit to get a nice smooth curve. And from these 2 smooth, I get my smoothed x and y coordinate, and these together give me then this curve. And in reality, this needs to be iterated, because how do I get the points in the right order? You see, in order to make this plot and fit this line here, I need to know these lambdas. And for this, I need to bring the points in the right order. And the way how the principal curve package does that, and I want to explain this in the next lecture in more detail, is if I have a curve, it first does a PCA. And and then it probably finds that this is the top principal axis, and then it projects each point on this principal axis and uses this as lambda, says this is lambda 0 and this is lambda 100, and puts the values in between. And then it has with that way a first guess for lambda, and then it when it can do this, use this guess to make our spline fits for the x and y, and then it might get a curve which then looks like that. And then, once it has this curve, it will again project each point onto this curve now, and in that manner get a refined thing, and it iterates this until convergence. So this is what principal curves does. And this is what we also used over there in our first thing here, where we looked at where we plotted this here, going from black over this to that. And now my problem that I had here was that I wanted a curve which goes from here to here. But what I got is a curve which goes from here to down here. And that explains why the cell cycle seemingly comes at the end. And this here is sort of and this stuff is somewhere in the middle, because all these things now get projected here onto the curve. And what I do to get around this now is I want to regress out the cell cycle. And that's the thing I now wanted to try with you. As we're not so many anyway, we can try this now slowly and see what to do with that. So to regress out the cell cycle means the following. You can imagine there are some principal components which go much into this and others don't. Let me show you by coloring by principal component. Here I have my so. So here, you remember this is what we get from the principal curve, which gives each cell a number. And I combine it here. I use the n-frame to make a table out of it. And now I take, let's say, the PCA embeddings. Here, these are the PCA coordinates. And I tell it to make a tibble out of that. And now I get here the cell and the tibble. And if I join that, I should now get a function with a with the x and y and these things. And now let me calculate. Sorry, that wasn't what I wanted to do. I wanted to make here a plot which is colored by pseudotime. Actually, you can also do this as an exercise while I write it. We want a plot where each point is colored by its value in principal component 1, 2, 3, and so on. But if you're now too lazy, I can also do it. So we take here, these are the UMAP coordinates, and these are the PCA coordinates. And we have to put them together. So I say I bind them with column bind. Put columns next to each other. Now we have a bigger matrix with 2 UMAP coordinates and 20 principal component coordinates like this here. And we can use this. We have to change it into a tibble, otherwise ggplot won't like it. And if I change it into a table, you have here these row names, and I have to turn it into a column. That's a property of tidyverse, that it always likes that. And now we have here this table here with all these columns. And now I can make a ggplot saying plot UMAP1 against UMAP2 and color by PC1. And if I do this, we can now see that PC1, for example, is a principal component that is strongly influenced by the cell cycle. You see it increases up to minus 30 in the cell cycle, and then it goes back to something like minus 10. PC2 it isn't that influenced by the cell cycle. See, it doesn't go much out. PC3 is also a bit less influenced. So one way how we could solve our issue is to calculate a distance, but ignoring the coordinates that go highly up here into the cell cycle. We could remove them from the matrix and recalculate our UMAP and our pseudo-time and see whether it looks better then. And there is one thing that the Serra people suggest that I now want to try out. I mean, we have 20 minutes left. Let's see if we get this to do. We have this cell cycle scoring function. And what it does is there is a list of genes which are known to change during the cell cycle. There are these here. Because there was this paper here quite a while ago in 2016 where they wanted to know, they were the first to realize the cell cycle problem and fall into that. And then they came up with the idea of making a list of genes which are known to be high in the cell cycle. So what they've done here is they made this list here, and you see these are all the genes which are usually high in the synthesis phase, in the S phase. So that's when the cell grows and duplicates its DNA. And these here are the cells which are high in the G2M phase, so in the growth phase and in the mitosis phase. Maybe, what is your majors? Are you your math people all? But when you get the cell cycle, and we have a look at the cell cycle. So this here is the cell cycle, how a cell cycles. You see the cell has to duplicate its DNA, then it pulls its DNA into two halves, and then it splits the cell into two halves. And often the cell cycle is put like this. The cell needs to grow, that's called the G1 phase, where the cell just produces enough material. Then comes the S point, where the DNA gets duplicated. Then comes the grow to thing, where the cell becomes ready to divide. And then comes the mitosis, which is the actual cell division, where the DNA is split into two packets, one with one copy, one with the other copy, and the cell splits in the middle. And you always have this cycling of going DNA synthesis, growth phase, mitosis or cell splitting, the next growth phases. And what we now have here is a list of genes which are always high, which peak in S phase, and a list of genes which peak in G2M phase. And we don't have a list of genes which peak in G1 because that's sort of a rest phase of a cell. And so what we assume is that all the cells which are high in the S phase are mark the beginning of a cell cycle, and those high in this G2M phase mark the next part. And now we get a bit into our biological issues. Here's the list of genes which are typically high in the synthesis phase, and here's a list of genes which are typically high in the growth mitosis phase. And as non-biologists, you don't notice that these these genes are all written in all caps, which marks them as human genes. While if you look at our count matrix, you notice that row names. You notice that these genes, they only have the first letter capitalized, which is typically for the mouse. So this is a list of genes. So what we have here given is a list of genes from humans, and we want to change it into a list of genes of the corresponding genes in the mouse. We could now try to simply change the capitalization and hope that it's the same, but in practice that probably doesn't work because the gene names are similar, but they do diverge. So we better look up in the official table that maps human to mouse genes. So what we want to get is this official table. And now this is a bit for you guys to learn how to work with this. There is this big database, the Ensemble dataset, produced by the EMBL, which you might know is the big institute up there on the hill. But this is actually not produced by the EMBL here in Heidelberg, but by their bioinformatics outstation in Cambridge, which contains all kinds of gene and everything about genes. And we want here the biomart, which is the database access. And I say, I want the gene database. I want the human genes. And then in the human genes, for each gene, I want to make a table which contains the name of this gene and its homologue. Here. A homologue is a gene which which corresponds in another species. So here we have the human gene name. It's the human gene name because I selected the human gene database. And here in M, I can now look for the mouse. You see there's a lot of strange animals. Lesser Egyptian jaguar. Anybody knows what that is? It's a funny little jumping mouse. I've seen it in the zoo recently. Mexican mouse. And here we have a mouse gene name. Now I've selected, I want a table with a human gene name and the mouse gene name. I can click here on results. And as you can see here, it shows us here a table of human and mouse gene names and their correspondence. Let me download this. No. Is it now? Yes, it has now downloaded this, called it Mart Export 4. So I read this. Read CS, no, what did I say here? TSV. Tilde slash tim slash downloads slash mouse ein Mart export 4. And here we have now our table. And now this is a bit how much R experience do you have? You worked a lot with R? Okay, then let's do, then we can use this as a way to do a bit of R thinking. So I now show you this slowly. So what I now do is I take this. Here you see I have saved this table as a TSV file, a text file where the columns are separated by tabs. I've loaded it with read TSV, and now I use this tidyverse verb to make this a bit nicer. First, I rename my columns. Human equals gene name. So this is the human gene name. And mouse equals mouse gene name. So this is just so that I know what's going on. Now it says here human, and here it says mouse. And I call this ortholog table. An ortholog is a what two genes are called orthologous if they are sort of evolutionarily related but in two different species. And now I can make my cell cycle here, ortholog table, and I want now only those genes where the human symbol appears in this CC genes $S genes. So these are our humans. And if I now split this, then you can see here, it has now given me here a list of corresponding mouse genes. And I can now pull them out, pull mouse. Maybe I first omit this NA here. You see for this gene, the ortholog was not available. So filter, Rufzeichen, ISNA, mouse. And now I have a table with two columns, but I only want the second column. So I say pull mouse. And now I have my S genes mouse. Mouse S genes. And I can do the same with the other gene set, with genes G2M genes, mouse G2M genes. Let's see if this works. Here I get a couple of genes, and you see here usually it's always the same CCNB2, but sometimes these numbers change. That's why I go through this official table. Now we have these genes, and now we can check where in our UMAP actually these genes are expressed. So what I now do, the first thing I could simply do is I take my log normalized data. This is this one here, layer data soy. I've never knew how to get this log normalized data out of Server, but I figured it out this morning. So here you see I have now my gene expression, and let's go in this thing only for the mouse S genes. And this is now the expression of these here. And let's add them up. Call sums. And now you see here I get the sum of all these gene expression values, just added up over them. And I could now plot, and now I want to plot my UMAP with this. How do I do this? I again make my embeddings Seurat, UMAP. So, and I say I make a table out of these things. So I take these embeddings, which is a matrix. So R makes this distinction between a matrix and a table. A matrix is just a 2D array of one type, and a matrix is something, and a table is something where each data frame can have a different data type. And in all these tidyverse stuff, you always need tables. So I say change this into a tidyverse table or a tibble. Again, row names should be the cells. Now I have this table here, where this is the cell, and this is UMAP1 and UMAP2. And then I say add a column, which I call S score. And now I can plot this. ggplot plus geom points aesthetics. X is UMAP 1, Y is UMAP 2, and color is S. And now we can see here these genes here, they are in the synthesis. These are the cells which are in the synthesis phase. I can do the same plot, of course, for the other one. G2M. Mouse G2M genes. Call is G2M. And I can see these are in the G2M. Now I can also see which way around the cells go through the cycle, because if I go back to my Wikipedia, you see S comes first and G2M comes afterwards. So it goes counterclockwise through the cycle. The cells come out here, go counterclockwise through here, and then they jump over here. Now the thing that they suggest is to regress out from each, what they now do is they have this scale data function, which we used already here in our, when we made our standard analysis. You remember what it does? It normalizes the data, which means divide each column of a count matrix by its sum and take the logarithm with this one pseudo count. Then find the variable features, so those genes with a lot of variance across cell. Then scale the data, which means what we said, which means from each gene subtract the mean and divide by the standard deviation, so that afterwards each gene has a mean 1 and standard deviation, mean 0 and standard deviation 1. And now, and then we use this for the PCA. Now, what R has suggested here, what Sörer suggests here is that we can redo this. Sorry, 2. But give scale data the extra list of variables to regress out, namely S genes and G2M genes. So let's give it these, I go with soy first in this cell cycle score function. And cell cycle scoring now wants this a list of S genes, mouse S genes, comma, mouse G2M genes. I am not 100% sure what this cell cycle scoring now exactly does, because, but it thinks it does what we've calculated here. It takes all these genes and adds up the expression of the genes in this list, because what we now get out, if I look at it here, soy $2, you see now there is this new thing S score. And my guess is that this S score is the same as what we've just calculated. Ctrl C. No. Yes. You see, this is what Sörer calculated, this is what we calculated. It seems to correlate well. There must be some subtlety slightly different. I'm not quite sure what formula it uses. We simply summed them up. Sörer's cell cycle scoring function must do something which is slightly more sophisticated than simply adding up the expression of his genes as we've done. But it seems to have the same effect. Anyway, once we have done this, what we can now do is hand this on to scale data and tell scale data regress these things out. S score, comma, how was the other called? G2M score. By regressing out, what we mean is when scaling a gene, it does a linear regression of the gene's expression. How do I correctly explain this? So for each gene, it makes a scatter plot of the strength of a gene against the G2M score. So let's say this is one of these scores, and this is the gene's expression. And now you can imagine maybe it looks like this, that this gene has, that this is a gene that has nothing to do with the cell cycle score. When the cells with high cell cycle score will have the same kind of expression than the cells with low cell cycle score. But maybe the gene is one of the cell cycle genes. Maybe one which was in the list, or maybe one that is missing in the list. In that case, the gene will be correlated to this S score. And then you get something like this, and you can make a regression line into that, again using linear regression and least square. And then you can subtract the fitted value, so subtract the y value of the line in order to, so that afterwards the line becomes horizontal. So this is what's called regressing out. Regressing out a score means make a least square fit to get a linear fit of the variable you're working on against the variable you want to regress out, and then subtract the values on the line, so that afterwards the correlation with this variable vanishes. And after the correlation vanishes, we should now get a new UMAP, and so all genes are manipulated that the part of a gene that is correlated with these two scores get subtracted, and this is then given to our PCA. So our new PCA coordinates should have no longer any information about the cell cycle. Not really, because you can imagine if the expression of this gene depends on the score in a non-linear way, then even if we subtract a linear fit, we will still be left with a residual, which the PCA will pick up, but the effect will be much weaker. So now I should get a UMAP in which the cell cycle is no longer that apparent. So let's see how our revised object looks like. Here's our new UMAP, and as you can see, it looks like this now. Let's make again the feature plots that I made before. Feature plot SOI2, what's it called? Aquaporin 4. You see here it starts. Here is the strongly expressed genes, the genes in the cell cycle. And you see, as I said, there is enough non-linearity in the data that our linear regressing out couldn't remove the cell cycle completely, but it's now much weaker, and now the neurons are properly, and now the whole thing is properly connected. And I can also add, so this double cortex gene that before was detached from the thing is now connected. I can also use the sleepwalk method now with our new revised Seurat object. And if I look at it now you can see that the cells that are within the cell cycle are now not very far away from the other stuff. So we have no longer the situation where we start and then go all the way far away, but now it looks more like this. And now this distance is much higher than this distance, and now the principal curve function should work properly. So let's try that. Again, I want to run my principal curve only through that part. So what I will do now is I again take out the lineage, do what I've done before here. And no, lineage clusters. So sorry if my code is slightly chaotic today. I probably should put this into a nice notebook. But, okay. the new lineage cluster labels, if I say label equals labels is true, when I think it puts numbers in it. No. What did I use last time? It gives me these labels. Label, not labels. So, now I can see which clusters are now in the lineage. It's cluster 6, 1, 4, let's take this 8 here to 0. I subtract my cells to only those here in this block, calculate my principal curve, and now I plot here the principal curve, stop, but with a new embeddings, with a new UMAP. What do I have here? PC lambda. This was also not the new UMAP. So now it seems to work better that it starts here and moves over there, but I'm not sure. Plus scale, color, VRDC. As you can see, it still hasn't worked properly. It somehow started over there. I must have not correctly. You see what the mistake I've done here. I've switched from this Sera object to my Sera2 object and copied the code, and of course always kept forgetting replacing Sera with Sera2. So right now it takes a while. Okay, now it has worked, but we start here and things increase here, go through the cell cycle and then move here. We still have a quite sudden jump here, but now it at least is in the right order. And with that now, we can now make plots of looking at specific things. For example, I can make a plot of this PC $lambda against any gene and can see how the gene goes up and down and along. But maybe we do this next week. So what I wanted to show you here, this was now a little bit of moving into different parts. We've got a bit into the biology. We saw the cell cycle genes. The genes for change weren't just random labels, but now we notice that they have different importances and that we can use this. This was now an attempt to just play with the data. I think for the lecture I will now prepare this properly so that we get it all nicely, that we write down exactly how the spline regression works, what the exact mathematics is behind it, where the spline basis comes from, where the Chebyshev polynomials are hidden in the whole thing, because you might have wondered what is this spline basis that I've drawn for, that I've made here? That one. What are these functions? I have to look it up again, but I think these are Chebyshev polynomials. They are constructed such that they partition the unity. So for each position, they add up to 1. So there's a lot of nice mathematics behind this spline regression. And I thought I'd show a bit how it works in practice before we look at the theory. Yeah, anyway, so let's leave it at that. I've recorded the thing in case any of the others want to look at it, but I don't know. Let's hope they turn up next time. Maybe I scared people away with too much Python last time. Let's see how this goes. Anyway, stop this recording.